My name is Dr. HANS. In 2000, I received my MD, started on a path of internal medicine and infectious diseases. And in 2017, I became a rare disease advocate when my son was diagnosed with SYNGAP1 syndrome. In 2021, I joined industry in order to work on rare diseases as much as possible. And I love talking about rare diseases, particularly when I have uninterrupted time, which I haven't for months. So today, I'm going to avoid the roller coaster news of rare disease that is so regular and stick to a strictly positive topic in that we just kicked off Q4 2025 and Camp4 Therapeutics dosed its first NHP in their GLP tox of a SYNGAP1 therapeutic. What did I just say? They dosed their first monkey, a non-human primate, in a toxicological study performed according to good laboratory practice. So I'm going to review the drug development process, and hopefully by the end of it, you have an appreciation of just how complicated it is, how hard the work is that everybody's doing that I'm astounded by, and hopefully you have a greater sense of what it takes and feel somewhat empowered in conversation and advocacy for the drug development process for your rare disease of interest. So I'm going to turn to this paper because it really had the best graphic I could find. It's not always easy to find something, perfect for use. And this was really a quite good illustration of the drug development process. So I'm going to use this and I'll include the link in the show notes. And it starts here, as you can see, with target discovery. And the idea is that this is where basic science, sometimes translational science, finds something new, right? The return on investment of basic science can be huge because when you discover that new thing, you're opening up a whole field of potential advancement to help that patient. What comes to mind is, being an intern on the wards, we had so much, cardiac disease, still to this day, obviously. And the 2B3A inhibitors were a new target with new drugs coming out that we were starting to institute to try to improve our survival rates for patients with myocardial infarction. Now, at least from the SYNGAP lens, we think back to 1998, where you've got the Mary Kennedy lab identifying SYNGAP, and you've also have the Rick Hucanier lab identifying SYNGAP. And this is key, but it's obviously not enough, right? You have this target, but a person naturally says, does it mean something for the human? Is it worth going after? If it is something that you could treat, will it result in benefit? And that's part of this target validation here. So the idea that this is a valid target in a human, that you will make a difference in their well-being if you go after it. As a topic, if you want to dive deeper into this and take a look at all the sort of categories of evidence for human target elevation, I'll direct you to this for a deeper dive, and I'll include the link. But for SYNGAP, the first sort of inkling was the paper by Jacques Michaud in the New England Journal of Medicine, going back to 2009. So 11 years from discovery to connection in a human. And here they have this cohort of kids, adults too, I believe, with non-syndromic intellectual disability. Non-syndromic, if you're across the room from someone, you say, Downs. Yep, I can see it. It's syndromic. Now, it's subtle, right? But SYNGAP1 has that syndromic label as well. But you wouldn't necessarily spot my kiddo from across a room unless he's doing his average, routine. But just from the physical attributes, it's a non-syndromic intellectual disability. And the lab here did deep sequencing and said, wow, 3 little girls have got a variant in SYNGAP1. And this is sort of the lead, right? It is a human illness. This gene matters to these kids. It's a start. It's only 3. But it begins that snowball rolling. And, after that, you then have, let's say, a mouse model, and it recapitulates the human disease, and it's considered a valid construct of the disease in humans. Now people are saying, okay, now we've got an animal model to experiment on. Fantastic. Does it still make a difference if you intervene? And now you've got evidence, again, out of the Rumball lab. If you correct SYNGAP, especially early in a mouse's life, you can restore their well-being, right? Their neurologic function. Later on as an adult, it's tougher, it's trickier, but even then, you will start to see some measures of improvement. So now we're building that case that it can be done in humans. Now we don't have, an approved drug to prove this beyond a shadow of a doubt, but enough companies are in the field that clearly people believe it. And I want to point out what I think was a big moment that I've not really heard beyond, just the, hey, Stoke is doing it. But I think that this IP, intellectual property submission in 2016, made a big difference because biotech, pharma has almost like PIs, right? Scientific private investigators that are snooping and trying to find the next target. And someone finds this and says, whoa, this is a really strong lab that is targeting SYNGAP. They know something. So let's look at it ourselves. And I think that this is fantastic. And if, you ever see Isabel Esnerez and Hugh Nash, thank them for this. Stoke is still trying to get to SYNGAP, right? They're concentrating on Dravet, and that's understandable. Small biotech, all the chips are in for Dravet, but this IP, I think, really meant a lot to our field. And you can find that here if you want to see the description. Beyond that, and let's go back to the graphic, this sort of builds that target validation case. Then, depending on what that company has for a platform, small molecule, ASO, antisense oligonucleotide, they're going to see what they can do for that target because it's a promising lead. I really got a kick out of a podcast with David Altshuler, the outgoing CSO, I believe retiring, our loss, well-deserved gain for him, describing the Vertex approach, where if they see a disease that has an unmet medical need, that they're going to think of the best way to address it and pursue that. And if they strike out, okay, we move on. Whereas other companies will say, what, we want multiple different shots on goal, mechanisms of action against a disease with unmet medical need. And there's pros and cons to each one, and I'm sure there can be a robust debate among, very, very well-experienced high-level thinkers on that, and that's not me. So when you decide to pursue that disease, and let's say it is a small molecule, and you can look at Gavin Rumbaugh's lab for an example of, here you can sort of see assay development and high throughput screening. And that's what they're trying to do. They've got a sort of repository of, I believe, 600,000 molecules at the Scripps down in Florida. And to run those 600,000 through a high throughput screen where, it's almost like a little twinkling of light in that assay says SYNGAP has been upregulated. You take that's your lead, and the medicinal chemists look at it and say, we can adjust it here, there. And I have to admit, going into pharma, I was looking forward to be in the room and watch medicinal chemists do their thing. And it is amazing. And they see that structure, and you know what, we're gonna fluorate the amide, and that's gonna improve the bioavailability and through the absorption. Or no, that looks toxic, that side chain. And clearly, pattern recognition, AI is helping them in their task. But this winds up generating your lead, right? And you're trying to optimize it and see how it does. And there's two obvious things that you have to show, and that's efficacy and safety. And that you can overcome, if it's not absorbed very well, or if it's metabolized and has an active metabolite, it's dumbfoundingly complex. You're on a global project team, and there's 20 brilliant people pouring themselves into this. And I really wish the public appreciated that, compared to some of the slanderous stuff you hear from other, opinionated people outside of the pharma world who have no scientific background or pharma experience. Anyway, what you see here is, efficacy. This would be, let's say, the SYNGAP mouse, and demonstrating that your treatment can upregulate SYNGAP, translate into behavioral improvement. This was something reported in Joe Anderson's paper, August 2024, where you can see improvement in, these rolling bar tests where the mouse is supposed to hold on, or elevated maze tests where they're exploring. But you also have to demonstrate safety. And this is an entire domain of preclinical safety. And you can see that here, and endless pages of FDA guidance or International Center for Harmonization and international expectations for what you need to demonstrate, starting off with, typically mice. And let me just say, this may be a little bit kooky, but I really think we need a national monument to laboratory animals. It's crazy to say, but think about it. We're talking about millions. Mice, monkeys. I know you're thinking it. Dogs too. Yes, I grew up with it. Lhasa Apso is a sibling, essentially. They make a sacrifice for the betterment of mankind, right? And it deserves to be acknowledged. Anyway, so you're demonstrating that, in particular in rodent species. Maybe a mouse is better, maybe a rat is better. It depends on the circumstances. Every project is so unique. Ultimately, you will need to demonstrate in a second species that safety. And it may be a dog. They've made a huge sacrifice for the hemophilia world. It could be a non-human primate, or NHP. And this is frequently the last step before you're getting into that clinical trial work, before you submit your IND, your investigational new drug application, where you're including this huge dossier of non-clinical, preclinical work, everything in the research world, and also your protocol for, let's say, first-in-human activities. And this is where Camp4 is right now. And it's very exciting. Still, you've got to manage your expectations, right? There are plenty of molecules that have failed in that last step in non-human primates. The idea that we've got a clear-cut genetic target that, SYNGAP, you can boost its level and it appears safe if you go too high. That is debatable. And I think that the key opinion leaders, the subject matter experts, debate this, right? Whether or not you can go high with SYNGAP. It's not necessarily a benefit to our kiddos to go above, that normal level. Let's say if my kiddo has 50% level, to double it. Does tripling it help him even more? That's what we have frequently called in medicine magical thinking, right? You give a week-long course of an antibiotic and patient goes home, chugs it, thinking that all at once is better. No, let's just stick to the plan here. So Camp4 has got their NHP, GLP talks going on. Good laboratory practice means that they're doing everything by the book. All their assays are validated. reproducible, reliable, that the handling of the monkeys is completely ethical, professional, documented, paper trail, audited, you name it. It has to be perfect. And then once those results are back, they'll be able to say, okay, here's what happened. Here's how much the SYNGAP level went up. Did the monkeys overreact? Was there any, they're going to slice those brains, just loaf them on a microtome, look at them closely. Was there any neurodegeneration? We've got to be able to explain why that happened. And, hopefully they see completely clean doses that they've selected. The top dose having an NOAEL, no observed adverse effect level, right? Clean as a whistle. And the FDA says, thank you so much for doing that. You may proceed with your first in human. Little bit of feedback, always. And then it's go time, right? By then, they will have their doses picked out. They will have their sites lined up, their informed consent. And you can see, Camp4 has already alluded to having their first in human open in the first half of 2026. Don't be surprised if it's June, because it does take months, right, to identify those sites, settle on a final list. They're already looking to have the IRB approved, the IND in hand, and to hit the go button. But that's how close we are. And when you consider from discovery in 1998 to 27 years, 28 years next, when we're likely to hopefully be dosing the first SYNGAPian, that's how much effort it has taken. It's measured in decades. It's getting faster. It has gotten faster. But think about it. All the people, all the disciplines in a pharmaceutical company or a biotech company like Camp4. I'm incredibly appreciative to my colleagues. I'm incredibly appreciative to the companies that are working on SYNGAP1. And, it's all about the patient, right? And they've gotten to know some SYNGAP1 patients. They know just how important this is. So stay tuned. Hopefully, Camp4 will publish their preclinical program that we can all learn from it. And we'll be seeing SYNGAPians dosed in 2026 and some early interim analysis and rumors that they're benefiting from it. And everything just goes smoothly. It's a lot to ask for, but keep your fingers crossed. Thanks for listening.